Okay, so prokaryotic organisms, that would be what organisms? Bacteria and archaea. Eukaryotic organisms, all the other four kingdoms, right? Animal, plants, fungi, protists. So you might remember that what's the big difference between eukaryotic cells and prokaryotic cells? The nucleus, and not just the nucleus, but any membrane-bound organelles, right? So eukaryotic cells are just much more complicated because we have all those membrane-bound organelles. We have the endoplasmic reticulum and the Golgi body and vesicles and vacuoles and mitochondria and chloroplasts and all those things that prokaryotic cells don't have. And there's a big difference in size because of that too, right? So when we're thinking about where DNA is located in prokaryotes, because we don't have a nucleus, that DNA is just floating in the cytoplasm. In eukaryotes, it's safe in the nucleus, a little more protected. There's also a difference in shape and number of chromosomes. So bacteria, for example, have one big circular chromosome. They can also have other, like plasmid, other circular chromosomes, but they have one main circular chromosome, whereas eukaryotes tend to have multiple linear chromosomes. So that varies from organism to organism. Like a fruit fly has 8 chromosomes. We have 46 chromosomes. Okay, so you can have a big range in number, but they're all linear. Another important distinction is that prokaryotes, their DNA is just DNA, whereas our DNA is wrapped around proteins, right? Those histone proteins. So if you remember back from the beginning of the unit, we weren't sure if it was the DNA from the chromosomes or the proteins from the chromosomes that were passing on traits. Right? Why is that an issue? Like, why do we care about that when we're thinking about replication and transcription? Why does it matter if it's wrapped around something? Well, what are we going to have to do to access it? We have to unwrap it. Yeah. So with prokaryotes, we can do replication and transcription without having to do a whole lot of extra stuff to our DNA. But eukaryotes, like us, we have to unwrap the DNA before we can open it up and copy it, or before we can open it up and transcribe it. Last really big difference on this slide is that prokaryotes do not have introns. So when they start transcribing a gene, it's straight code right away. Okay, they don't have to do all the extra processing and splicing that eukaryotes do. So if you think about the time that it takes for a prokaryote to go from DNA to RNA to protein, and a eukaryote to go from DNA to RNA to protein, what do you think the difference is going to be there? Go ahead and talk about that in your groups really. Saying that's so good, like it's not the eukaryotes that have to be done. Why can't we all just take prokaryotes? Very simple life. Here in the world. I'm not near that, but I'm cold. See, like, divide it. Okay, I think you're cold. So, probably, hopefully, pretty obviously, it's going to be much quicker for prokaryotes to do anything with their DNA, to copy it or transcribe it, than it is for eukaryotes. Because if we want to make a protein, we have to unwrap the DNA, and then we have to edit the mRNA, and then the mRNA has to leave the nucleus. If you're a prokaryote, you don't have to unwrap it. As soon as you make the mRNA molecule, it's made. You don't have to edit it. And it doesn't have to leave the nucleus. So for us, it takes about an hour to go from DNA to protein. It takes a much shorter time, like within seconds. Well, maybe not seconds, in minutes at least, for bacteria to make a protein. Because like we mentioned, the tRNA, like everything's random motion, right? Everything has to run into each other. So if you have to leave the nucleus and then run into each other, it's just going to take that much longer. And so you need to find a way. Basically, it's much faster for prokaryotes. Because you don't have to process the RNA, and you don't have to worry about it leaving the nucleus. So because there's less physical separation, it also takes less time. Something along those lines. I'm not going to test very heavily on this. It's just kind of a synthesis of what you know about different cellular structures. So last slide on this topic. This is showing under a microscope here. This is prokaryotes. So because the DNA is just floating in the cytoplasm and there's no mRNA editing, the transcription and translation processes are like simultaneous. So sorry, I know this is kind of low for some of us, but you can see here, this is kind of the key. This strand here is the DNA molecule. Off of this, an mRNA is being copied. And as soon as that mRNA, part of that mRNA is copied, a ribosome is going to attach and start translating. Okay? So in prokaryotes, those transcription and translation processes are happening right away, simultaneously. I think this also does a good job of showing that you can have multiple ribosomes copying the same RNA sequence. So as soon as one ribosome attaches and it starts passing through, then another ribosome can attach. So we're going to make multiple DNA sequences with the same mRNA, or sorry, multiple proteins with the same mRNA sequence. But besides that, it's still the same code. So, like, if we put a human gene in a bacteria, we would have to make sure that it didn't contain the introns, but it would make the same protein. So, like, if we wanted bacteria to make insulin, as long as we put in a gene that codes for just the exons of insulin, it would make insulin for us. Okay, so the genetic code works in the same way. The mechanisms are slightly different, as you can see in this case, but so that's all I didn't want to spend a whole lot of time on that, but any questions on the differences between prokaryotes and eukaryotic cell translation? So a little bit more important, and this will be on the test, is some information on mutations. The lab we're going to start tomorrow is dealing with various different mutations. So first of all, what's the definition of a mutation? A change in specifically DNA. Yeah. So in order to be a mutation, it has to be a change in the DNA. Now, if the DNA changes, your mRNA code is also going to be different. Actually, your protein chain is going to be different. Although not always. So there's two categories of mutations we're going to talk about today, one on this slide, one on the next, and some similar but different results that can occur from both types. Also, and again, sorry for the size of this diagram because I know it's probably really hard for some of you to see it. But we have a term called wild type. Wild type is like the normal sequence. So whenever you see the word wild type, that's like the normal common DNA sequence versus the mutations, which are going to be the new or different versions of the DNA code. So first type of mutation we're going to talk about is called so Penguin says, when do mutations affect the next generation? So what kind of cells would these mutations have to happen in order to pass them on? Your reproductive cells, yeah, your egg and sperm. So we haven't talked about meiosis yet this year, but these mutations would have to happen in our egg or our sperm to get passed on. If they happen in any of our body cells, that still can affect us, but it's not going to get passed on to the next generation. So it's something to think about. First category is point mutation. So this is a change along one singular point in the DNA. And so this is really a substitution. So we're doing a single base change. So if I have a C, I'm going to switch it with a T or an A or a G. So in all of these situations we're going to take a look at here, we're swapping one of the bases. So in this case, we're swapping a C for a U. In this case, we're swapping a G for an A. And in this case, we're swapping an A for a U. Okay, so in these cases, we're just switching out, substituting one base for another. And that's going to cause a point mutation. These point mutations are very situational in their effect on the protein, as is much of biology, very situational. But sometimes they cause what we call a silent mutation. What do you think a silent mutation is? Where it doesn't affect the amino acid sequence, and therefore doesn't affect the protein. It does affect the DNA. The DNA changes, the mRNA changes, but the amino acid sequence is going to stay the same. Okay. Why can that happen? The third base wobble, yeah. So remember, there's some redundancy in our code. So if the mutation happens to occur where we're substituting a third base for another third base, in a lot of cases, that's going to give us the exact same amino acid. And so if we have the same amino acid sequence, we're going to have the same protein, right? Same secondary, same primary, same secondary, same tertiary, same quaternary structure. So these mutations are interesting because they can kind of help us give a timeline for evolutionary change, because mutations happen at a fairly constant rate without affecting, in this case, without affecting the proteins that get made. So even if there's not like visible changes in populations over time, they're still going to pick up these silent mutations. The next resulting possibility is a missense. So this is a mutation that will in fact change the amino acid sequence. It will only change one amino acid in the sequence, right? But it will change one. And depending on the properties of that amino acid, the new amino acid, there can be more or less change in the structure and function of the protein. Right? So you might remember some R groups are hydrophobic and some are hydrophilic, and that affects the way the protein folds. Our lab tomorrow on Monday will kind of go into that a little bit. The last possibility could result in a nonsense mutation, and that's when it's going to cause a premature stop codon.
So do point mutations have to change amino acids, or is that just a general change?
It's just a change in the DNA base. And so like silent mutation is not going to affect the protein. The amino acid stays the same. And the codon still codes for the same amino acid. So some mutations don't cause any effect. Right? Some mutations are going to completely destroy the functioning of a protein. Some mutations might actually cause a new function in the protein that we didn't have before. So everything is situational. This kind of depends on the situation. Okay? I'm going to try and get this picture a little bit bigger on another slide. It's not going to get much bigger, but. So here's our first substitution. We're looking at C for U. In the wild type, the codon that results codes for glycine. In this case, it's still coding for glycine. We're just changing the third base, and that part of the code is redundant. So again, that's a silent mutation. There's no change in the amino acid sequence. Here, the first base in a codon gets swapped. So instead of coding for glycine, it codes for serine. And again, depending on how different the properties are on that amino acid, that's going to affect how differently the structure of the protein is. And then here, when we make that swap, it accidentally codes for a stop codon. So we're going to stop making the protein early. As you can imagine, that's almost always just a loss of function, right? It cut the protein off, especially at the beginning. The closer you are to the beginning, the more likely you are to just have a completely non-functional protein. Yeah.
So what is the point mutation so that the protein never starts?
Yeah, then that would definitely be a loss of function too, right? If it started even before the start codon. Well, I mean, remember, this is just one protein. So depending on what that protein does, it may or may not affect you a lot. Okay. There's also the fact, and when we get into genetics, you don't have just one copy of most genes. You have two copies, one from mom and one from dad. So even if one of your copies is mutated, it doesn't work. Sometimes the other copy will work. So there are a couple mechanisms that we have to kind of protect us from that. Okay, so here's a mutation that causes sickle cell anemia. Go ahead and talk about this in your group. What kind of mutation is that?
Say it's yeah, fast. It doesn't stop. Yeah, it doesn't say stop, and it didn't say the same thing. We can now say QED.
I don't think it could be that bad. If you look ahead in here, oh, we should learn how to do it today. It's it's fine. You guys have more time.
It's true. We also did not know
Now, like from learning it, the first
Okay. So in this case, it's definitely a point mutation because we're just substituting one base for another, right? So we have a point mutation. It's also considered to be a missense, yeah, because the amino acid that it's coding for goes from being glutamine to valine. And these two amino acids have very different properties. So glutamine is hydrophilic and valine is hydrophobic. And so that causes the proteins to clump all together and make that sickle shaped cell. Okay, so sometimes one little change can have a big change in the protein structure and therefore a big change in the protein function and affect the whole organism. And we'll talk, depending on how far we get this year, there's some really cool treatments for sickle cell. Okay, so that's one class of mutations. The other class we're going to talk about today are called frame shift mutations. So remember, we set our reading frame by starting at the start codon, right? And that tells us what 3 bases go into which codon, okay? So if I do a frame shift mutation, like I get rid of an amino acid, that's going to shift what letters go together in the codons, right? So now instead of AAG, it would be AGU, and then everything after that is going to be shifted, right? The whole code gets shifted. So frame shift mutations are going to cause a shift in our reading frame. So which letters get read together in the codon? And so there's really two ways we can create a frame shift. We can either delete bases and take them out, or we could add bases in. That's going to mess up all the reading frame too, right? So when we do these frame shifts, we're going to change all the codons downstream of it. And again, we're going to either do that by insertion, adding bases, or deletions, losing bases. Sometimes you lose whole chunks.
Should figure out the next slide.
But the results use a lot of the same terms as the point mutation, so we'll go through this. Again, this is situational. Obviously, where would this mutation cause the most change? If it was the beginning of the gene or the end of the gene? The beginning, because again, everything after is going to get affected. The only case where it would not affect every single amino acid after is if we delete or add exactly how many bases? 3, right? Because then we just be either adding in a whole amino acid or losing a whole amino acid. But the frame shift, that all the letters would still, after that would still be read together in the same codons. Okay. So there is a little bit of course, losing or gaining an amino acid is also potentially very damaging to a protein. So if we focus on this, we can see, first of all, we have a deletion happening here. So if we think about our terminology from our other mutations, if we have changes in amino acids, what do we call that? Missense. So this is just a lot of missenses happening, right? So the term that they use in the diagram here is extensive missense, okay? So extensive changes to the amino acids. Yeah, so this can also result in a nonsense. This can be a nonsense too. Yeah, so in the frame shift, we create a stop codon. So to get missense, we can get a nonsense, or we could just get an insertion or deletion of amino acids if it happens at exactly 3 bases in or 3 bases out.
Isn't like extra bases, like let's say one gets removed, so there's just like 2, and that doesn't make the whole codon. Do those just kind of like do nothing, like once before the start or after the stop?
So if it's before the start, it's not going to cause anything, right? But if it's after the start, then everything's going to get shifted.
Do these like do nothing? I guess. Yeah, like if there are just 2 extra, do those just do nothing?
Well, if we put 2 extra in here, like we're still going to shift the reading frame, right? It would only be if we put a some multiple of 3 that we wouldn't disrupt all of the reading frame. Okay, that makes sense. So We'll talk, maybe I'll do a do now on that tomorrow, but like I said, we're gonna start a lab tomorrow, so we probably will move the desk back so you can look at the I don't know. I sometimes don't know what the best thing to do is.
Me all the time.
So.
You don't have to log
Where else is that?
okay, so maybe I used the wrong words there. They happen at a fairly small rate.
That's all I get.
They can also happen
Okay. Oh, my bad. What's up, JC?
Right there. Make I
Having like presents.
Can I put myself in there?
Sure.
You see Queen Elizabeth?
Nice.
Yeah.